guys, welcome you back to my channel Biomics. Hope you are doing well and practicing for your exams. So in the last video we were discussing some questions regarding the basics of DNA replications which are asked in CSIR net. So in this today's video we will discuss about the questions which are asked in DNA replication in eukaryotes. So let's get started with the video guys. This is the first question. स्पीड ऑफ डीएनए रेप्लिकेशन इन मैमल्स इज 256 बेस पेयर पर सेकेंड. मैमल्स में जो प्रेजेंट है उसका जो डीएनए का रेप्लिकेशन जो प्रेजेंट है उसका स्पीड है 200 बेस पेयर पर सेकेंड. सो फॉर रेप्लिकेशन ऑफ 3 × 10⁹ बेस पेयर डीएनए इट विल टेक वेरी लॉन्ग टाइम बट इट इज कम्प्लीटेड विद इन फ्यू आवर्स. ये बोल रहा है कि बहुत टाइम लगेगा अगर 256 बेस पेयर पर सेकेंड के हिसाब से हमारा डीएनए रेप्लिकेशन हुआ. सो व्हाई इट इज कम्प्लीटेड इन ए वेरी शॉर्ट टाइम? First option is most of DNA is introns, high amount of replicative DNA, repetitive DNA, multiple origin of replication, only small portion is replicated in one cycle. So the correct option is multiple origin of replication. अगर वो intron हो या exon हो, it does not matter. The thing is that DNA is replicating. So it is not the correct option. High amount of repetitive DNA. अगर repetitive DNA भी present है, it needs to replicate the repetitive DNA. So this is also not the correct option. So the correct option is multiple origin of replication. Now the second question. Semi-conservative mode of DNA replication is in eukaryotes can be experimentally demonstrated using. ये बोल रहे हैं सेमी कंजर्वेटिव मोड कैसे डेमोन्स्ट्रेट करेंगे. द फर्स्ट ऑप्शन इज द करेक्ट ऑप्शन, दैट इज ब्रोमो DU लेबलिंग. नाउ नेक्स्ट, with which one of the following statement is correct with reference to replication in eukaryotes? सिंगल ओरिजिन एंड कंटिन्यूस रेप्लिकेशन नहीं, इसका मल्टीपल ओरिजिन हो रहा है. सो दिस इज इनकरेक्ट. मल्टीपल ओरिजिन, यस इट इज मल्टीपल ओरिजिन एंड सेमी डिसकंटिन्यूस रेप्लिकेशन, यस दिस इज द करेक्ट स्टेटमेंट. यहाँ पे दिया गया है मल्टीपल ओरिजिन एंड कंटिन्यूस रेप्लिकेशन. कंटिन्यूस रेप्लिकेशन नहीं होता है, यहाँ पे होता है सेमी डिसकंटिन्यूस रेप्लिकेशन. नेक्स्ट, सिंगल ओरिजिन एंड सेमी डिसकंटिन्यूस रेप्लिकेशन. यहाँ पे सिंगल ओरिजिन नहीं होता है, यहाँ पे होता है मल्टी ओरिजिन. ओके, सो नाउ द next question. In eukaryotes, shortening of chromosomes for end is prevented by. The answer is telomerase. Telomerase एक enzyme है which is preventing the end level replication, or we can tell that it is preventing the chromosome for shortening of ends. Next, in order to study the role of telomeres in DNA replication, genetically engineered mice were prepared where the genes for telomerase RNA was knocked out. यहाँ पे क्या किया गया? टेलोमेरेज का जो स्टडी कर टेलोमेरेज एंजाइम का स्टडी करने के लिए यहाँ पे कुछ एक्सपेरिमेंट डिज़ाइन किया गया जिसमें कि आरएनए का माउस का जीन का नॉकआउट किया गया जहाँ पे है टेलोमेरेज RNA. When cells from this knockout mice were taken and cultured in vitro, they proliferate even after 100 cell division, which is quite unlikely in case of human cells. Which one of the following is the correct reason for this question? The first option is human and mice are fundamentally different with respect to the requirement for telomerase enzyme in the content of DNA replication. ये तो दोनों तो eukaryotes है, we cannot tell. This is the correct statement. Next, in vitro mice DNA becomes circular due to end-to-end chromosome fusion and does not require telomerase for DNA and replication. Third option, mice have very long stretch of telomerase DNA sequence compared to that of humans. Yes, this can be the probable statement that mice can have a long stretch of DNA as compared to the mammalian or human cells. Okay, so C option is the correct statement. Next, during replication, the RNA primer is degraded by the 5' to 3' exonuclease activity of. यहाँ पे पूछ रहा है कि RNA प्राइमर का जो डिग्रेडेशन हो रहा है, who is doing that? It is DNA polymerase. No, DNA polymerase cannot do. Next is topoisomerase 2B. What was the work of topoisomerase 2B? It was helping in preventing from supercoil. So this is also not the correct statement. Next is FEN1, which is known as flap endonuclease. Yes, this is the correct option. ये जो है flap, इसका नाम है flap endonuclease, but it is doing the work of exonuclease. Okay. Next question. फॉलोइंग आर सर्टेन स्टेटमेंट रिलेटेड टू यूकैरियोटिक डीएनए रेप्लिकेशन। यहाँ पे कुछ स्टेटमेंट्स दिए गए हैं जो कि है यूकैरियोटिक डीएनए रेप्लिकेशन के बारे में, एंड वी हैव टू फाइंड द करेक्ट स्टेटमेंट। द जीनोम ऑफ मल्टीसेलुलर एनिमल कंटेन many potential origin of replication. Yes, this is the correct statement. यूकैरियोट्स में मल्टीपल origin of replication present रहता है. During early development, when embryos are undergoing rapid cell division, origin sites are uniformly activated. Yes, they are uniformly activated. Pulse, next option. Pulse chase technique is used to label sites of DNA replication. Yes, this technique is used for labeling of DNA replication. Next, fourth option. The rate of elongation of different DNA chains during genome replication varies drastically. No, it does not vary drastically. जो elongation का step है, it has certain, there are certain specific base pairs. If there is DNA involvement of DNA polymerase 1, 2 or 3, there are certain numbers of base pairs which will be added, and that during the process of elongation there is no drastic change of the genome replication. So the A option is the correct option for this question. Okay, now next question. Eukaryotic DNA polymerase a high as tightly associated primase activity but moderate processivity. ये जो DNA polymerase जो है, it is having tightly associated primase activity but moderate processivity. Processivity इसका moderate है लेकिन इसका primase activity ज़्यादा है. Given below are 4 statements about leading and lagging strand synthesis in eukaryotes. यहाँ पे कुछ statements दिए गए हैं जो कि है leading as well as lagging strand के बारे में. Which one is true? The first statement is both leading and lagging strand are synthesized by DNA polymerase alpha. Moderate processivity is essential to maintain the fidelity of replication. No, this is not the correct option. ये जो है DNA polymerase alpha, it is firstly synthesizing, but after that the leading strand is being synthesized by DNA polymerase epsilon, and DNA lagging strand is synthesized by DNA polymerase delta. Okay. Next, entire leading and lagging strand are synthesized by epsilon and delta. Eukaryotic replication is primer independent process. No, it is not a primer independent process. It is also dependent upon the primer. Only the lagging strand synthesis need primer and synthesized by DNA polymerase alpha. No, first thing is that lagging strand only does not need primer, leading strand also need primer. The second thing is that lagging strand is synthesized by DNA polymerase delta. Okay, next, primer for both the strand are synthesized by DNA polymerase alpha. Yes, it is being synthesized by DNA polymerase alpha. DNA polymerase alpha synthesize the primer, and there is polymerase switching for epsilon and delta. For epsilon, it is leading strand is synthesizing the epsilon, and delta is for lagging strand. Okay, so this is the correct option. So D statement is the correct option. Next, in eukaryotic replication, helicase loading occurs in all replicators during this phase. All the replicators are being loaded. It is in the G1 phase. Here the replication, here everything is loaded but they are not activated. Okay, they are activated during the S phase, that is a synthetic phase or the synthesis phase. Telomerase, a RNA protein complex which completes the replication of telomeres during DNA synthesis, is a specialized RNA dependent DNA polymerase. Yes, is a RNA dependent DNA polymerase. It is a DNA polymerase but it depends upon the RNA because its telomerase enzyme, it is working as a RNA. Telomerase, a protein RNA complex, has special reverse transcriptase activity that completes replication of telomeres during DNA synthesis. Although it has many properties similar to DNA polymerase, some of them are different. Which one of the following properties of telomerase is different from that of DNA polymerase? यहाँ पे क्या हो रहा है, टेलोमेरेज का भी कुछ प्रॉपर्टीज है, डीएनए पॉलीमरेज का भी कुछ प्रॉपर्टीज है. ये पूछ रहा है कि दोनों में डिफरेंस क्या है. फर्स्ट स्टेटमेंट इज दैट टेलोमेरेज रिक्वायर्स ए टेम्पलेट टू डायरेक्ट द एडिशन ऑफ न्यूक्लियोटाइड्स. ये भी ये जो प्रेजेंट है ये टेलोमेरेज का भी प्रॉपर्टी है और डीएनए पॉलीमरेज का भी सेम प्रॉपर्टी है. इट रिक्वायर्स ए टेम्पलेट फॉर डायरेक्टिंग द न्यूक्लियोटाइड. टेलोमेरेज जो प्रेजेंट है, सो दिस इज नॉट द करेक्ट स्टेटमेंट बिकॉज यहाँ पे क्या पूछ रहा है, यहाँ पे पूछ रहा है कि डिफरेंस क्या है. नेक्स्ट, टेलोमेरेज कैन ओनली एक्सटेंड ए 3' एंड ऑफ डीएनए. ये जो डीएनए पॉलीमरेज जो एंजाइम है ये भी क्या कर रहा है, इट इज एक्सटेंडिंग द 3' ऑफ डीएनए. सो दिस इज ऑल्सो नॉट द करेक्ट ऑप्शन. टेलोमरेज डज नॉट कैरी आउट लैगिंग स्ट्रैंड सिंथेसिस. यहाँ पे बोल रहा है कि जो टेलोमरेज जो प्रेजेंट है इट इज नॉट वर्किंग ऑन लैगिंग स्ट्रैंड. ये जो प्रेजेंट है ये सिर्फ लीडिंग स्ट्रैंड में ही काम करेगा. बट डीएनए पॉलीमरेज क्या, डीएनए पॉलीमरेज बोथ लीडिंग स्ट्रैंड में भी काम कर रहा है और लैगिंग स्ट्रैंड पे भी काम कर रहा है. So this is the difference between the DNA polymerase and telomerase. So this is the correct option. Telomerase acts in processive manner. ये DNA polymerase is also working in a processive manner. Now the next question. Question number 12. In eukaryotic replication, priming of DNA synthesis and removal of RNA primer is catalyzed by. ये बोल रहा है कि eukaryotic replication में डीएनए का जो प्राइमर जो होगा वो कौन सिंथेसाइज कर रहा है और आरएनए प्राइमर का कौन रिमूव कर रहा है. इट इज़ बाय डीएनए पॉलीमरेज अल्फा. डीएनए पॉलीमरेज अल्फा जो है वो प्राइमर का सिंथेसिस कर रहा है. इट इज़ वर्किंग एज ए प्राइमेज. और आरएनए प्राइमर जो प्रेजेंट रहता है when the primase is working and the primer is being synthesized, यहाँ पे जो आरएनए प्राइमर रहता है उसको रिमूव करने के लिए जो एंजाइम चाहिए वो है FEN1. Okay, now the next question. In eukaryotic cell, replication initiation from a replication origin occurs only once per cell cycle, and S-phase CDK play a vital role in the regulation of DNA replication. In budding yeast, a protein complex known as ORC is associated with DNA replication origin during G1. However, origins fire only once at the beginning of S phase. DNA replication does not start in G1 phase. ये बोल रहा है कि जो G1 phase में DNA का replication क्यों नहीं हो रहा है, S phase सिर्फ S phase में ही क्यों हो रहा है. Now let's see the statement. MCM helicase are inactive in G1 phase. हाँ, जो MCM helicase जो present है, they are being loaded in the G1 phase, but they are not active in the G1 phase. Okay. So this is the correct option. Are inactive. ये जो present है, it is a inactive. It is loaded in the G1 phase, but they are activated in the S phase. So जैसे ही A option हमारा correct हुआ, then we can eliminate these two option. Next, spindle checkpoint is active in G1 phase. नहीं है, spindle checkpoint ये active G1 phase में नहीं हो रहा है. This is the incorrect. DNA polymerase is not recruited in G1 phase. Yes, DNA polymerase is not recruited in the G1 phase. It is being recruited in the S phase. Next, ORC and initiation factor CDC6 and CDT1 do not recruit MCM helicase to the site of replication initiation in G1. It does not recruit MCM helicase. No, it is MCM helicase is recruited in the G1 phase, but the activation is being done in the S phase. So this is the incorrect option. So our answer is A and C. Okay. Next. Following statements were made about human mitochondrial genome. ये जो कुछ statements है mitochondrial genome के बारे में पूछा गया है. ठीक है. The replication of both the H and L strand is unidirectional and begin at specific origin. ये बोल रहा है heavy और die strand का जो है it is unidirectional और ये specific origin से begin हो रहा है. Yes, this is the correct option. जैसे ही A correct statement है, we can eliminate these two options. So A is the correct statement here. Next, majority of the mitochondrial gene encode for protein products. No, it is majority of the mitochondrial gene is active or no. This is the incorrect option. Though the mitochondrial genome is extremely compact, the genes never show any sequence overlap. नहीं, जीन का overlap हो रहा है during the H strand and L strand synthesis. There is overlapping of this genes, or the sequence is being overlapped. The CRT loop the CRD loop region of mitochondrial genome exhibit triple stranded structure. Yes, this is the correct option. जब कभी भी लैगिंग स्ट्रैंड एक हेड स्ट्रैंड रहेगा, एल स्ट्रैंड रहेगा और एक स्ट्रैंड जो है एल स्ट्रैंड का या हेड स्ट्रैंड का सिंथेसिस होगा, ड्यूरिंग दैट टाइम इट फॉर्म ए ट्रिपल स्ट्रैंड स्ट्रक्चर. सो डी ऑप्शन इज द करेक्ट स्टेटमेंट. जैसे ही बी इनकरेक्ट हुआ, वी गॉट द ऑप्शन बी इज द करेक्ट स्टेटमेंट. नेक्स्ट ई. Transcription of mtDNA starts bidirectionally from a common promoter region in the CRD loop region and continues around the circle. Yes, this is also the correct statement. Now the next question. Given below a few statements related to DNA replication. यहाँ पे DNA replication के बारे में कुछ statements दिया गया है. The first statement is replication in eukaryotic chromosome from the origin is initiated multiple times in each cell cycle, while it is initiated only once in each cell cycle at the origin in bacterial chromosomes. ये जो है ये multiple times origin नहीं हो रहा है. It is being initiated only at a time, but it is having multiple origin in case of eukaryotic chromosome, and in case of bacterial chromosome in case of eukaryotic chromosome it has multiple origin, but in case of bacterial chromosome it has a single origin, but it is initiated at only a particular time. So this is the incorrect option. और यहाँ पे incorrect option ही पूछा गया था, so this is the incorrect option. Now यहाँ पे A statement के साथ D statement भी present है. Like first we will see the D statement. Rolling circle mode of replication produces multiple units of original molecule. Yes, by the help of rolling circle mechanism of DNA replication there are formation of multiple molecules. Now the B statement. Improper reinitiation of replication in a eubacterial chromosome is prevented by hemimethylation status of bacterial origin. Yes, it is being prevented by hemimethylation. अगर fully methylated है then it is ready for DNA replication, but if it is hemimethylated it is resistant to origin. That means यहाँ पे origin नहीं होगा अगर ये DNA का कोई सेगमेंट hemimethylated हो तो. ठीक है. सो यहाँ पे ये स्टेटमेंट भी करेक्ट है. नेक्स्ट, DNA polymerase 3 is a major replication polymerase responsible for de novo synthesis of both leading and lagging strand. Yes, ये DNA polymerase 3 जो है both it is synthesizing the leading and lagging strand, and ये this is the major DNA polymerase. So now let us question. The following statements are made with reference to the replication of DNA. यहाँ पे कुछ स्टेटमेंट्स दिए गए हैं जो कि है replication of DNA के बारे में, and we have to find the incorrect option from this. The eukaryotic counterpart of the bacterial beta-clamp protein is proliferating cell nuclear antigen (PCNA). ये बोल रहा है कि beta-clamp जो present है it is equal to PCNA in case of eukaryotes. Yes, this is the correct option. और यहाँ पे पूछ रहा है कि incorrect option कौन सा है. So next, mutation in active one of the subunit of the MCM27 helicase negatively affects the initiation of DNA replication in eukaryotes, but has no effect on elongation of the replication fork. No, this is not the correct statement, because ये जो MCM27 जो है it is working as a helicase. अगर इसका mutation हो गया then it will not allow for the unwinding. अगर unwinding नहीं होगा तो how the process of DNA replication, how it will elongate. So this is the incorrect option. Next, all DNA polymerase responsible for replicating the eukaryotic genome catalyzes DNA chain extension in a DNA template dependent manner. नहीं, all DNA polymerase नहीं, there are different type of, except DNA polymerase 1, 2, 3, there are other different DNA polymerase, other different polymerase, like it is DNA polymerase, zeta DNA polymerase, alpha. Okay, there are certain other polymerase which work in a template independent manner. So this is the incorrect option. Now next, the FAN1 proteins play a, so जैसे ही B और C incorrect हुआ, so this is the correct statement. Now next question. DNA given below are set of enzymes of column A and enzyme activity of column B. Okay, so यहाँ पे दोनों कॉलम में कुछ स्टेटमेंट्स कुछ दिए गए हैं जिसको कि हमें मैच करना है. So first is DNA topoisomerase 1. DNA topoisomerase क्या करेगा? It will help in single strand nicking. ये जो है single strand nicking करेगा, तो A is 4. Next, topoisomerase 2 double strand break and ligation. ये जो B जो present है it is for 3. Next is polymerase epsilon. What polymerase epsilon does? It help in the leading strand synthesis. ये जो C जो present है, it is 2. Next is polymerase delta. Polymerase delta, it is needed for the synthesis of the lagging strand. और lagging strand में क्या formation होगा? Lagging strand में formation होगा Okazaki fragment. So polymerase delta, it is synthesis of Okazaki fragment. D is 1. So यहाँ पे option A is the correct statement for this. Now the next question. The following statements were made about mitochondria. Which one of the option represents a combination of the correct statement? यहाँ पे correct statement पूछ रहा है. The D loop of mitochondrial genome is required for replication but not for the regulation of transcription. नहीं, वो D loop जो present है वो transcription के लिए भी responsible है और replication के लिए भी responsible है. तो so this is not the correct option. जैसे कि ये नहीं है, so option A and option C are eliminated. The L strand of mitochondrial genome possesses more cytosine. Yes, ये जो present है, it is containing more cytosine. B is the correct option. Next, in plants, most mitochondrial tRNA are encoded by nuclear genome and then imported into mitochondria. Yes, this is the correct option. Next, cyclohexamide inhibit protein synthesis by mitochondrial ribosome but does not affect eukaryotic cytosolic राइबोसोम. नो, दिस इज नॉट द करेक्ट ऑप्शन. अगर वो इनिवेट करेगा तो दोनों को इनिवेट करेगा, अगर नहीं किसी को भी नहीं. ए, सम ऑर्गेनिजम्स हैव बीन फाउंड टू कैरी लीनियर माइटोकॉन्ड्रियल डीएनए. यस, सम ऑफ द ऑर्गेनिज्म कंटेन्स अ माइटोकॉन्ड्रियल डीएनए. सो द करेक्ट ऑप्शन इज बी, सी एंड ई. ओके, नाउ द नेक्स्ट क्वेश्चन. इन Saccharomyces cerevisiae, DNA replication is tightly controlled. A DNA should replicate once per cell cycle. Choose the incorrect statement regarding why the cells does not replicate their DNA in S phase. यहाँ पे पूछ रहा है कि incorrect statement find करने के लिए why the cells do not re-replicate their DNA in S phase. Re-replicate क्यों नहीं हो रहा है उसके बारे में पूछ रहा है. Pre-replicative complex, pre-RC, remains bound to the DNA in the S phase and does not allow the re-replication. Next, assembly of pre-RC is inhibited by CDK activity. Yes, this is the correct statement. Assembly of pre-RC is initiated at the end of mitosis at the early G1 phase of the cell cycle when the APC activity is high. Yes, this is also the correct statement. CDT1 that help in the recruitment of MCM protein in the G1 phase is inactivated by geminine. Yes, it is inactivated by geminine in the S phase of cell cycle. So this is also the correct option. So the incorrect option is first option, option number A. ओके, यहाँ पे हम देख सकते हैं MCM27 जो कि हेलिकेज है, इट हैज बीन एक्टिवेटेड बाय DDK एंड CDK. ये DDK और ये जो दोनों जो प्रेजेंट है, दे आर द काइनेसेस. दैट मीन्स इट विल गिव द फॉस्फेट ग्रुप, इट विल ऐड द फॉस्फेट ग्रुप. और ये CDK जो प्रेजेंट है, CLD2 और CLD3 को भी एक्टिव कर रहा है. ओके, आफ्टर द एक्टिवेशन ये जो प्रेजेंट है DBP11 और अदर पोलीमरेजेस लाइक पोलीमरेज इप्सिलॉन, जीन्स एंड CDC5 भी आकर यहाँ पे अटैच होता है. एंड आफ्टर दैट CLD3 and SLD2 are being removed and here it forms the CMG complex. यहाँ पे CMG complex present होता है. Next, after this the polymerase alpha or the primase is being loaded over here. Okay, after that DNA polymerase delta is loaded, due to which there will be synthesis of the primer and there will be synthesis of the leading as well as the lagging strand. Okay, now the next question. The following statements are made about telomeres. Telomeres are believed to shield telomerase from the cell's DNA repair machinery, preventing them from being recognized as double-stranded break. Yes, this is the correct option. टेलोमरेज के पास टेलोमरेज बाइंडिंग प्रोटीन्स होता है जो कि सील्ड करेंगे टेलोमियर्स को फ्रॉम द सेल्स डीएनए रिपेयर मशीनरी, प्रिवेंटिंग देम फ्रॉम being recognized as double-stranded break. Okay, so the next option, telomerase in human cells are repeats of TTA GGGGG sequence that can extend up to 150 kb, which are replicated by the axon or the TERT in actively dividing cell. No, this is not the correct option. In differentiating cell, telomere is inactive, leading to shortening of telomeres over hundreds or cell division, damage to ends of chromosome, and eventually apoptosis. In differentiated cell, telomerase is inactive, leading to, yes, this is the correct option. The persistence of telomerase activity in several cancers allow the cells to continue to proliferate. हाँ, अगर telomerase में कुछ प्रॉब्लम है, अगर telomerase continuously वर्क कर रहा है, then it will allow the cancer cell to proliferate. So option D is the correct statement. Next, in eukaryotic cell, DNA replication is restricted to S phase of the cell cycle because eukaryotic cell में DNA replication S phase में क्यों restrict हो रहा है? The DNA polymerase is prevented only in the S phase of the DNA polymerase is present only in the S phase of the cell cycle. Origin ORC recognizes origin only in the S phase. ये only जैसे ही only देखेंगे हम delete कर सकते हैं, इसे omit कर सकते हैं. MCM helicase get activated in the S phase of the cell cycle. Yes, this is the correct statement. MCM helicase जो present है, it is being loaded in the G1 phase, but it is activated only in the S phase for the synthesis or for the replication to take place. Next, MCM helicase get activated in the G1 phase of the cell cycle. No, it is not activated. It is being loaded in the G1 phase. So this is the incorrect answer. These were some of the questions of CSIR Life Science Net of the topic DNA replication in eukaryotes. In the next video, we will discuss about some of the questions regarding the DNA replication in prokaryotes. And if you like this video, please do like, share and subscribe. If you are subscribing my channel, I will be motivated to do such videos. Thank you.